First up is Brian Blanchard. Brian is a partner with Scotian Halibut, which is one of Canada's foremost marine hatcheries. Been with the company for quite some time, and we've had the privilege of knowing and working with Brian for many, many years. And Brian's going to talk about how it is that genomics is important to Scotian Halibut. So over to you, Brian.
Thank you very much, Steve. Hello, everyone. I was doing fine until I had to follow that video. I have no idea how I'm going to make it as exciting as she did. We're just doing a very small piece of work here in Atlantic Canada. I'm with Scotian Halibut. I've been the general manager for over 12 years, and we've been involved in start to finish bringing along a technology and a business in a small community in Nova Scotia, in Clark's Harbour and in Woods Harbour. And it's been a lot of work and a lot of difficulty. And one of the shining stars of why we're where we are today is because of the genomics project and our plurigene project that we did in conjunction with Genome Atlantic, Genome Canada, and Genome Spain. I don't have a whole lot of time, so I was going to cover a brief look on broodstock development. And I put up this as a timeline, and if you look back, it actually even goes further than the 1997. It really goes back into the early 1990s. But these are some of the accomplishments that we worked on to make broodstock reliability in our hatchery more effective. And photothermal adjustment, basically we were able to change and manipulate light regimes and temperature regimes so that we could spawn year round. We also looked at using implants, hormonal implants, to help time spawning with the females. We also, a project that looked at developing all-female populations because females grow faster than males. That work was going on. Also, egg assessment work when we started using very finite diagnostic tools to figure out lipid composition in eggs to see if we can improve through nutrition. We also did, that rolled into the nutrition aspect of how do we manipulate or improve egg quality through nutrition means. And then we moved into the Genome Canada work, the plurigene project. And what that did is it really gave us a huge new tool or toolbox of information that we could apply to the breeding program. And if we drop down to the bottom where we have the broodstock and bio security and domestication program. We started that in 2004, and if you see, I'm trying to represent it as best we can. It was a set program, very linear, and it was based on traditional breeding methods. With the Genome Canada work and the linkage development and the map, we were basically able to expand that program phenomenally, and that we can basically start taking traditional breeding programs and turning them into much more tangible science-based breeding programs. To bring a female halibut along to maturity is actually a 12 to 15 year process. So if you make mistakes, we can't go back in time and fix them, and I'm not going to be around long enough to correct it going forward. So the real tool that we have that's making a difference is the genetic markers and the genetic tools that are available to us. I put this one, I love this slide. These fish are all brothers and sisters from a cohort from 2004. And the fish that's circled, if you look at that fish compared to all the rest of them, there's a reason why that fish is larger. We don't know exactly why it is, but locked in its genes is the information that we need from the breeding program to be able to best pick these fish going forward so that we're not making mistakes. We don't have the luxury of time as a private company to make mistakes. And in fact, if we had known that it was a 12 to 15 year time period to bring a fish along, there's not an investor in Canada or the world that would have supported any of the work that we've done. And so we've kind of snuck through over time, and hindsight is great, but this is the reality. And it's not only for our species, it's actually for most species, except the generation times. When we look at salmon, we have to remember that DFO and hatcheries were going on in this country for easily 100 years. And because of that 100-year history, domestication had already been worked into the population. All of the fish that we're working with were collected from the wild in the mid-90s or late 90s, and so we really only have one crack at getting this right, and genomics is the tool that helps us get it right. Just so that you get an idea, this is what a female halibut would look like. This would be the egg collection piece. We actually manually strip everything, so we actually have an ability to pick and choose who we're using. And I put this one up here. This is a poster that we did from the Plurigene project, and with it we were able to send out, the bottom part is the map that we created with the project, but we sent this poster out to the world. There's 3,200 copies all over the world that went through trade publications, and I've been in Spain, I've been in Norway, and I've been in Iceland, where these posters have been up on company walls. And if you look on the bottom, it's hard to see, it has all of our partners that were involved in this. And it was a tremendous, tremendous accomplishment. And what it did, and still has, is it says that Canada is the first to have gotten the genome correct, and this is the tool that we use going forward. And the rest of the world basically stood back and said, okay, you figured it out, no problem. And now we're doing partnerships, and we're working with Norwegian companies who are buying juveniles from us, who are actually coming to us now for stock and eggs as part of our breeding program, because there's a recognition that we're 7 years to 10 years ahead of everything that they could start today. And because of that, and because of the genetic tools, we've basically positioned ourselves in Atlantic Canada in an unbelievably good place. The hard part is just keeping the working capital, keep the whole company going. But the reality is we have this 10-year lead, which is backed by science, and that science comes from the genome aspect of it. I just wanted to give you, this is a lovely slide. This is what's actually going on when we talk about why are we doing halibut. If we look at the Pacific halibut fishery, these are the quotas. The quotas are dropping like rocks. There's pressure to increase quota from the fishermen. There's a lot of pressure to reduce quota from the advisors. There's discussion about reducing the size of age of capture. The end result is 11,000 metric tons is what the current recommendation for the quota is, which means that there's nearly 20 million metric tons of product not in the marketplace, which is a monumental opportunity for Canada, a monumental opportunity for anybody who's growing halibut. And when we look at Norway, Norway has about 1,200 metric tons of harvest annually of farmed product. Canada, I can't even put a number up there because we were at about 200 metric tons. We've since declined to about 60 metric tons because of lack of working capital. 90% of our sales are actually to Norway right now. We're trying to reverse that trend, but part of it is just the growth that's going to happen. And again, I love these nighttime shots. The market for us is right next door. You know, this is what we're trying to do in Atlantic Canada. We're trying to produce a product that does no longer exist from a wild source, that's basically next door and looking for it. And so that's one of the success stories that Scotian Halibut's been able to talk about. And the reason we are where we are today, and it's not just because this is a group, it is because our breeding program has advanced better than everybody's, and we've been able to put science along with that breeding program, which basically says we really do know what we're doing with our decision making. So thank you very much.